Sorry to talk about infection. So today, first thing that I want to discuss is what determines the virulence of infection. We can call it determinants of virulence/severity. Here we can look at two sides. One side that determines virulence, of course, comes from the pathogen. So on the pathogen side, we have infectious dose, hence how many pathogens you actually get into you. We have portal of entry. I just want to put them in a specific order. And virulence factors, which we will discuss separately. So infectious dose, that parameter reflects the amount of microorganism you get in. For some microorganisms, this factor does play a role, for some it doesn't. For instance, for our beloved coronavirus, there are no data suggesting that the amount of virus actually changes anything in the course of infection. Portal of entry, I'm going to be using coronavirus left and right. Portal of entry, if you apply it to the skin, the coronavirus, nothing's going to happen. Okay? If you put it in your gut for the urinary tract, nothing's going to happen. Okay. And virulence factors, we will discuss what they are. Just I want to say a few words about the host side. So on the host side, what can influence? First thing that comes up to mind immediately is age. Of course, immune status. If the person is highly suppressed. Genetic susceptibility. Some people are known to be more sensitive to certain infections. Previous exposures. Yeah, you want, if you want to, you can work that. If I'm not forcing, but if you want to, feel free. It's totally fine with you. Previous exposures like the vaccine or previous encounters with that pathogen. We can come up with a gazillion other factors, like we can say the nutritional status, malnourished people, more susceptible, most likely. But it all kind of falls into one of these broad categories, okay? So let's address the specific category of virulence factors. So the first set of virulence factors that I wanted to discuss would be exoenzymes. Remember, enzymes that are produced and released out of the cell. Now, I want to make a little remark here. When I'm going to be discussing two main categories of virulence factors, which are exoenzymes and toxins, but it doesn't mean that that's it. Antibiotic resistance is also considered a virulence factor. Presence or absence of capsule, slime, or flagellum, it's all virulence factor. I just want to focus on these two. There are 4 types of enzymes, which you must know, as almost everything with this particular unit, you need to be able to match the description of the term. What they do, what is the enzyme? So first type is glycohydrolase, which breaks down carbohydrates. Second type is protease, breaks down proteins. Nuclease, which breaks down nucleic acids. such as DNA or RNA. And finally, this Rabies. That breaks down phospholipids. Hence, again, you don't need to memorize specific examples, okay? Be able to match the description of the enzyme with the target. You clear so far? Good? Okay. Now another category of virulence factors are toxins. Now, toxins can be further divided into two subcategories, which are endotoxins and exotoxins. So endotoxins, or lipopolysaccharide, are produced exclusively by gram-negative bacteria. They are found essentially in the outer membrane of the cellular envelope. Remember we talked about gram-negative envelope, membrane, cell wall, outer membrane. LPS is in the outer membrane. The unique features include, first of all, endotoxins have really high LDP, which means they are unlikely to kill you. Very unlikely. And they will give you help in terms of the symptoms, non-specific symptoms. So, fever, headache, muscle ache, joint ache, you generally feel like that. It's a generalized inflammatory response that produces that kind of sensation. Does that make sense? And importantly, they are very stable, specifically thermally stable. And what else should I say about that? Oh, they do not form immunological level. So when you are exposed to endotoxin, your immune system, of course, responds, but it does not form any antibodies or T cells, no immunological. Good. Exoenzymes can be produced by both gram-positives and gram-negatives. Okay. They're relatively low LD50, so some of them are, in fact, botulinum toxin that we mentioned before is the most toxic natural compound that was discovered. They produce very specific symptoms. Okay, and they're not too stable, not too stable, and they form immunity. Okay, we clear here? Now, I want to highlight that exotoxins can be divided into three categories. You only need to know, again, our categories. I have a table in the lecture notes with all the categories and examples. I'm going to ask you, what is the mechanism of action for Phagocytosis toxin right now? I mean, I will eventually, not in this year. Or what is the mechanism of action for cholera toxin that is in the table? But you should be able to match the description of the toxin with one broad category. So these broad categories are intracellular targeted exotoxins, membrane targeted, and superintelligent. So intracellular targeting toxins, and it's just not from immunity in general. So intracellular targeting toxins inhibit or influence certain specific processes in the human cells, like botulism toxin inhibits the release of the exocytosis of acetylcholine, or cholera toxin messes up the cyclic A and B signals. So very specific processes. Membrane targeted toxins target membrane like streptolysin, Streptococcus pyogenes. Okay. Breaks down membrane. And superantigens overstimulate immune systems such as toxic shock, syndrome toxin, Staphylococcus. So far, make sense? Okay, good. Now, we're going to talk, I'm going to leave this place for some infection-related terms, because I want to feed as much as I can, so it all stays on video. You can photograph it if you want to. But next thing that I want to talk about are stages of disease and address some aspects of those disease stages. And when I say disease, remember, it's a microbiology class, so we're talking about infections. I appreciate other diseases, but we are here for infections. Actually, I will correct this. Not disease, infection stage. We, ironically, we're kind of doing it backwards, because the virulence, the whole pathogenicity, the pathogenesis situation, it happens at the very last thing, right? Because the first stage of disease will be adhesion. So for adhesion, we need to know mechanisms for two categories of microorganisms. For bacteria, you have to know, and by know I mean just know what those 5 mechanisms are, through which bacteria can adhere to the surface of the human cells. It will be proteins called adhesins, fibrillate, bilateral, capsule, and slime. Now I want to make, now look at this. If microorganism adheres to the surface more efficiently, it makes it more virulent. Does that make sense? Does it make it a virulence factor? Like if the microorganism has fimbriate, so it makes it more adhesive, makes it more virulent, therefore fimbriate is the virulence factor. All those 5 things are essentially virulence factors. Anything that facilitates it. For viruses, adhesion is mediated by receptors. That's a really important concept. And it explains certain features. For example, mice cannot be infected with poliovirus because they simply don't have a receptor. Conventional mice are not susceptible to SARS-CoV-2 infection because they do not have proper human receptor. Okay, does that make sense? Next category, I'm going to put it here, invasion. Not much to say about bacteria. Invasion in regards to bacteria refers to extra or intracellular. So, for instance, while Staphylococcus aureus can exist in the extracellular space without getting into the cells, many bacteria prefer to get into the cells. Many do both. Salmonella typhimurium can cause extracellular infection, but can also infect cells per se. Does that make sense? Now, viruses, as you may remember, always intracellular. They are obligate intracellular parasites. Okay. Does that make sense to you? Okay. Next step is probably the most fascinating step in the entire field of microbiology, and most microbiologists, I would say, that study human disease, they study this, which is immune evasion. Okay. If you search for a position in microbiology, it's all about host-pathogen interaction, which is actually elimination. In the lecture notes, I have a whole page on the immune evasion. Okay, I'm not going to recite all those mechanisms in details because they are, many of them are self-descriptive. Actually, I think it's no, yeah, I don't see how it's printed. Yeah, I think it's, that's it. Yeah. So, for instance, the mechanism of complement inhibition. Well, that's complement inhibition. That's it, right? You see what I'm saying? Inhibition of innate immune response. That's it. Self-explanatory. Things that I want to highlight, and I'm going to say e.g., meaning that, for example, capsule. And the fact that I list these here, please understand, I'm not giving them extra importance. I just want to explain what it means. Capsule prevents phagocytosis, proteases. That can be tricky. They kill antibodies, destroy antibodies. Phagocytosis, bodies, leukocytins. They kill white blood cells. Those are special proteins. Okay. Leukocytins. Oh, molecular mimicry. Now that's probably my favorite. In case of molecular mimicry, microorganism kind of wears an enemy uniform. Well, we are the enemy. So schistosoma, small follicles of the blood fluid, can cover itself with human blood proteins. Does that make sense? So human immune system, when it cannot recognize schistosoma because it is covered literally with human proteins. You clear? Good? Yes. That brings us to another interesting conversation, which will be about the stages of disease. I'm going to put it right under fusion, invasion. So, stages of disease. Now, I'm going to draw a little graph here. And once again, I'm going to use our beloved coronavirus as an example. So the blue graph will represent how your disease progresses. Now, if we talk about COVID-19, disease progresses as such. You get infected. Not you personally, please. It's my habit of saying you. I'm not referring to any other person. You get infected, you don't know about that. And we have some people who can attest to that. So you don't know about that, you're already infected. That's going to be our incubation period. So blue y-axis shows the severity of symptoms. You have no symptoms. Then at some point you start developing symptoms, but they are not too bad yet. That's going to be prodromal period. Okay. And then it hits you hard. How hard? Depends on the patient, but it hits you hard. In some people, it's going to be full-blown. A friend of mine had myalgia, arthralgia, loss of sense of smell, severe migraines, no fever whatsoever, no respiratory problems. I know some people had really bad respiratory problems. I know some people who were like just tired. Okay, all spectrum. The point is, you have after prodromal, so 1, 2 prodromal, 3 is the disease period. Now what happens here? What happens here at the peak? Well, 2 things for any disease, and I don't want to get morbid, but that's the reality. The patient either dies or gets better. Right? There are some other possible outcomes, not for coronavirus. Gets better. That's the decline phase, and then you get into convalescent phase. Okay? So we can put them on the list here. First is incubation period. Second is prodromal. Disease, decline, and convalescent. Okay? Now, since we're talking coronavirus, okay? Well, let me think about, okay, let's start not with the coronavirus. Let's add one more virus to the equation, common cold. So purple is common cold. Common cold, what you're going to have? You're going to have the replication more or less coinciding with the disease. Does that make sense? So on the peak, when you like sniffling and sneezing and feel like crap, you have high titers of the virus, you can isolate. Okay, good. For corona, the situation is very different. You have a peak of replication early, then you have a decline. So the highest, what I'm trying to illustrate, look, I'm not going to ask you specifically about coronavirus in the exam, because nobody could predict this one. That's right. Question? But the point is, the peak of replication of a microorganism does not have to coincide with the highest severity of infection. Am I clear? So you do have a graph in the lecture notes where they do coincide. It's fine. It's fine. But it's not always the case. Don't take it as a sort of a final solution. Does that make sense to you? Are we clear? Now, in regards to the disease, I want you to clearly distinguish between signs and symptoms. Symptoms are subjective, signs are objective. So if I walk into a fever clinic in Vienna and I say that I have myalgia, arthralgia, and headache, I give them my symptoms. And no one can check those symptoms. If I walk in there, start vomiting on the floor, they take my temperature and it's 104, those are signs that something is wrong. I objectively vomit and I objectively have fever. Does that make sense? So, and for instance, nausea, and we presume that we trust the patient, but nausea is a symptom. Vomiting is a sign. Bowel upset is a symptom. Diarrhea is a sign. I don't have time to go into, but there are like there's clinical tests how to determine if feces can be classified as diarrhea. Yeah, we can talk during the break. It's not a super important, but there's. Signs, symptoms, stages. Okay. We are getting to fill that little space here. I'm going to talk kind of related to the stages of disease, like what can happen. The example that I provide on this image, I'm going to call it types of infection. Okay. And the first type of infection will be acute. Okay. So, replication of the pathogen and disease. Now, please understand, I use the word disease because it's short and convenient. When I say disease, you have signs and symptoms. You say, okay? So what we deal with here in this example are the examples of acute infection. Influenza, coronavirus, common cold, salmonellosis, like traditional garden variety salmonellosis are examples of acute infection. An infection can be chronic. In chronic infection, you also have replication and disease. The difference between chronic and acute is that chronic is a long term. Many people consider chronic disease as being milder. There's nothing mild about it. If you think about disease like tuberculosis, acute tuberculosis, initial infection is really mild. Chronic tuberculosis is extremely severe. There are, for instance, giardiasis, intestinal infestation with a protozoan called Giardia lambda. It doesn't really have any acute phase. It hits you hard, and 3-4 months, if you do not treat it, it's a chronic diarrheal disease. It's really bad. I had a student who had giardiasis, and the student said she wouldn't wish it on her worst enemy. It was so bad. Okay. Now, another type is latent. We talked about latent infection, okay, like herpes virus infections, papillomavirus infections. We generally consider there's no active replication, and there is no disease. Now, virus, or usually virus, stays under the radar of the immune system in the cells, doesn't replicate, does not cause any disease, can reactivate and cause symptoms from time to time, but not permanent. Does that make sense? But probably the most fascinating type of infection, which we, quite frankly, don't know the exact mechanism of, the persistent infection. In persistent infection, host does have replication, but has no disease. A couple of classic examples. Well, the classicest example, sorry for butchering English language, the most famous, I would say, example is that for Mary, a cook in New York, who was infected with salmonella, never had any symptoms, but she was shedding salmonella persistently for decades. Okay. It is known that cytomegalovirus can cause persistent infection in children, where children shed it for months without having any symptoms whatsoever, or signs. Polyomavirus called BK virus can show the same pattern of behavior in pregnant women. Pregnant women are slightly immunocompromised, so they can shed BK virus in their urine in pretty large quantities. no symptoms, no signs, no damage ever done. Like nothing. Does that make sense? So again, you have to be able to match the description of the infection type to the infection type. Okay. Now, we kind of mentioned brief importance of entry, but we never really talked about the routes of transmission. And this is what I'm going to do right now. I'm going to give you an overview of routes of transmission of infectious disease. Now, in this case, obviously, I am talking about communicable diseases. Things like peritonitis or infections that, for instance, if you, God forbid, scrape your foot really bad in the forest and the wound becomes infected. I mean, not really, you're not going to give it to another person. So we're talking about more sort of exposure. So there are two types of transmission routes. There's a direct route, there are direct routes, and there are indirect routes. So in case of direct routes, we are first talking about contact transmission. This is the transmission that requires close contact between the people. Contact with persons' biological substances, like feces or vomitus or sweat. Example of contact transmitted disease would be Ebola. Although we put STDs in a separate category, technically speaking, it is a contact transmission as well. Direct transmission between two people because they get in contact with physiological, biological substance, which is mucus, the mucosal membrane. Does that make sense? Next, again, coronavirus. We have respiratory transmission. Now, in respiratory transmission, there are two submodalities. One submodality is droplet, and another airborne or aerosols. So what's the difference? Droplet transmission occurs when you sneeze, cough, speak, sing, exercise. Basically, your strenuous respiratory activities, like the ones I just listed, generate respiratory droplets, large droplets. Does that make sense? But there's therefore physical distancing. I hate the term social distancing. Physical distancing. Even if I will take off my mask, chances are droplets that I generate when I speak, they will not reach you. I am fine. Can some of them do? Yes, absolutely. This is why we wear masks, because the main modality of transmission for coronavirus is large respiratory droplets. When people say, and I am going to be very crass here, when people say that masks do not contain virus, they are either hypocritical or morons. Viruses are so small, even paper masks do not contain virus. We do not release virus. We really release droplets when we speak, or very, very fine aerosols when we breathe. Can aerosol go through the cloth mask? Sure thing. Can it go through N95? Last time I checked, there are not very many hospital infections in the surgical rooms. So, no, aerosols cannot. That makes sense? Virus, oh yes, sure, it can go through anything. But again, we do not produce virus. So those people say these things? I really won't. Never mind. Since it's recorded, I don't want to say anything. And vector transmission. Vector is an animal in overwhelming majority of cases. We are talking about arthropods, mosquitoes or ticks. And I try to generalize, but direct human transmission, to my knowledge, occurs only by mosquitoes. Well, more general, Diptera, the family Diptera, it's family order, Diptera, okay? Mosquitoes, some flies. So for instance, if malaria-infected patient gets bitten by a mosquito, and this mosquito bites a naive patient and transmits the infection, that's the direct vector transmission. Same goes for yellow fever. Does that make sense? Now, not all infections that are transmitted by mosquitoes can be transmitted directly between people. West Nile virus cannot. It's a zoonotic infection. Mosquito bites a bird, and then a human. If I will have West Nile, there will be a swarm of mosquitoes, and they will all bite me, and they will attack you and bite you a gazillion times. You won't get infected. Okay? Indirect routes. Fomites, inanimate objects. Now, if we use coronavirus as an example again, droplet transmission is number one. Aerosol transmission is probably number two. Well, not probably. Number two, I would put this as being more important. Because when we together, we usually don't just breathe, we also speak. Fomites play minimal role in the transmission. All this bleaching of every single grape that we bought at the grocery store, I didn't do that. Made no sense. Okay. Vehicles. Not objects, but material. Realistically, there are two ways of vehicle transmission, food and water. Okay, does that make sense? Unless someone's trying to consume food. Right? So, but I mean, realistically, when you drink milk and get salmonella, that's vehicle transmission. When you drink water and get giardiasis, vehicle transmission. I'm going to put aerosols, but I will explain what it is. The virus called Synnombre, a hantavirus that causes severe acute respiratory distress. It is shed by deer mice, and it is shed in the deer, not deer, but deer. I know my pronunciation. D-E-E-R. I know it. They shed it in the urine and feces. Urine and feces is dry, get aerosols. Human inhale that aerosol. Does that make sense? Get infected. So that's indirect aerosol transmission. And I'm going to put here in little parenthesis vector. And since I'm about to expand on vectors, you will see why I'm not hesitating, but it's not like traditional, it's not very common transmission route. It's a different type of vector. So, what types of vectors do we know? Vectors can be biological, or mechanical. So what's the difference between biological and mechanical vectors? The biological vector, basic biological vector, microorganism reproduces in the vector. It's best exemplified by mosquito example. Mosquito bites a person with malaria. Malaria parasite reproduces, goes through a certain life stages of development in the mosquito, and then mosquito bites in a person, transmission. Person with yellow fever gets bitten by a mosquito. Yellow fever virus replicates in the mosquito's salivary glands. Mosquito bites another person. Bird with West Nile gets bitten by a mosquito. West Nile replicates in the salivary glands, bites a person, gets West Nile. Does that make sense to you? Replication. In case of mechanical vector, there is no replication. It's purely mechanical transmission. And look, this is what I'm referring to. Mechanical vector. Fly lands on a pile of feces, then lands on fluid. Whatever was on that pile of feces, whatever bacteria there were, they get transferred to fluid. There's no replication, just carries it away. Does that make sense? You've got the difference. Transmission routes, vectors. Okay. A small bit that I want to, I put it on the board. There is a list of 5 categories of carriers. We consider, when we say a word in terms of our epidemiological study, when we say a word carrier, we mean a human. Okay? Does that make sense? We're not talking about dogs, raccoons. People use it, but strictly, when we're going to speak strictly, when I say a carrier, I refer to a human. So mosquito that bites me and gives me West Nile is not a carrier. Does that make sense? Okay. 5 types of carriers are incubation, right? Before symptoms onset, chronic carrier, convalescent carrier, asymptomatic carrier, and passive carrier. So the difference between asymptomatic and incubation, in case of asymptomatic carrier, the person is not going to get disease at all. Typhoid Mary was a classic example of asymptomatic carrier. She had no symptoms. Okay. If I am not sick, but I'm infected already, I'm going to be incubation. Okay, when your friend comes over after recovering from diarrheal disease, and they find and they sit with you, but then a couple of days later you develop the same diarrheal disease, they were convalescent carriers. Does that make sense to you? Okay, I'm trying to think. Okay. Couple more terms that they're there, just give an example. Host versus reservoir. Host is someone who gets disease. Reservoir is where infection, usually where infection resides normally. Without reservoir, microorganism is gone. Again, the best thing to do is use examples. You're going to soon, I think, experience nausea when I say word coronavirus. But coronavirus, initial reservoir was most likely bats. Hosts are we, humans. Are we now a reservoir as well? Yes, we are. We have become both hosts and reservoirs for infection. Since cats can be infected, they can get sick. They can. Are they hosts? Potential hosts? Yes. Can they serve as a reservoir? We don't know. That makes sense? Good example of host versus reservoir interaction. Rabies. The reservoir are wild animals, skunks, raccoons, foxes, whatnot. If we wipe them out, hypothetically, rabies is gone. Okay? Are they hosts? Yeah, they are. Are we hosts? Accidentally, yes. If you wipe out humans, though, rabies is not going to disappear. Does that make sense? Example where hosts and reservoirs are the same species, and the infection is basically restricted to those species, measles. Humans are the reservoir. Humans are the host. You wipe out humans, measles is gone. Understood? Okay. Now, let's say a few words about some epidemiological things, then I will talk briefly about Trypanosome. I don't know if I'm going to put it on the board because that's not really, everything is in there, just things to kind of focus. This I want to put on the board. So, few epidemiological terms. First term is incidence. Incidence is the number of infections per population per time. Numbers that I'm going to give you are completely made up, like totally made up. Deal? Okay? So I'm going to tell you that there are 3 cases per 100,000 people, 3 cases of coronavirus per 100,000 people per day in the United States. Okay, that's incidence because it's number of cases in the certain population per time. Can you say that there are 90,000 cases in the entire population of the United States per day? You absolutely can. You give a reference to population. Usually, population parameter is 100,000 people. Does that make sense? Because otherwise, you can't compare a number of cases in, say, Monaco and the United States. There will be prevalence. Number of infections per population. No time this time. The prevalence of coronavirus in the United States, as far as we know, is what? 9 million cases per the entire population. Good? Now, I'll address some morbidity. The trickiest term here, because morbidity is often defined as number of disease per number of infection. So, kind of strictly speaking, if we have 100 infections and 100 people get sick, that's disease with high morbidity. Unfortunately, there is a tradition, and we can't do anything about it, to use the word morbidity in more qualitative terms. For instance, we tend to say that diseases that are more severe are highly morbid. Does that make sense? So for instance, we say, oh, Ebola is the disease with high morbidity. People get really, really, really sick. And common cold is the disease with low morbidity because people don't get really sick. Yes, 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 we use that. Make sense? And lastly, mortality. That is a really interesting parameter because strictly speaking, it is defined as number of deaths per number of infections. Okay? This is also known as IFR, infection fatality rate. Am I clear? Now, the tricky part is that we do not diagnose all infections. Some of them are asymptomatic. Some people don't go to a physician. Make sense? And interestingly, we don't fix all the deaths because we don't know, like a person dies at home, never been tested. That's it. We will not know that it's a death that is related to a particular infection. So both a numerator and denominator of that ratio, that fraction, can be deflated, or depending on how you diagnose, if your tests are not good, can be inflated. So what we use here in the coronavirus program, we could use slightly different parameter, which is case fatality rate, CFR. Now, case fatality rate calculates the number of deaths divided by the number of confirmed cases. You see the difference? And now I consider it being my sort of public service to fight misinformation. So I run some simple arithmetic. Case fatality rate for coronavirus in the state of Ohio is a little bit less than 3%, like 2.9, okay? That makes sense. Number of deaths over the number of total infections. Confirmed cases. If you would do the same math for the state of Iceland, where they did an absolutely astonishing testing, like insane testing, you're going to get case fatality rate at about 0.5%. Okay? If you run the same calculations for the state of Singapore, and I did that yesterday, you're going to get case fatality rate at 0.04%. Does that make sense? If your healthcare system is impeccable, Singapore, and you diagnose the living daylights out of the population, knowing of all infections possible, okay? You have really small number of deaths, a huge number of infections. Your CFR is super low. If you decide, screw diagnostics, we're just going to do bare minimum, but people keep dying, then your CFR is going to be fast to move. Does that make sense? Now, not on the board because sporadic, endemic, epidemic, endemic. Sporadic cases are popping from time to time. They're not really common. Rabies in the United States is sporadic. Malaria in the United States is sporadic. Does that make sense? Endemic. Disease is fairly common in a certain region. Coccidioidomycosis in the southwest region of the United States. Like really southwest, like California, Nevada, Utah. Okay? Really common. Influenza is endemic like everywhere, right? You see what I'm saying? That's endemic. Epidemic, above the regular level. You can have an epidemic outbreak of influenza. We expect a certain number of cases every winter. If it's triples, that's epidemic. Okay? Or you can have an epidemic outbreak of cryptosporidium when you expect 1 and you have 200. That's already an epidemic, more than you expect. Pandemic worldwide. Okay? Now, before we get to Trypanosome, a few words about what kind of questions you can expect. All kinds of match the term to the description. Also, I will ask you plenty of questions on characterizing an infection. Let's take example of West Nile virus. I tell you, West Nile virus, people get West Nile virus when they're bitten by a mosquito, and before that mosquito has to bite a bird, okay? So you say, okay, this is a zoonotic infection transmitted by biological vector. Okay, that makes sense? Or Yellow fever can be transmitted from person to person or from monkey to person by a mosquito, and virus replicates in a mosquito salivary gland. You tell me, that is zoonotic, possibly zoonotic infection that can be directly transmitted between humans by a biological way. Like you pick the programs. Or you get salmonella from milk. Milk is daily. You leak a faucet handle and get noroviral diarrhea. Faucet handle is foam. Does that make sense? Things like that. In regards to the Trypanosome, there are two species that I talk about, what you need to know, where they are. It's solder in the nose. Trypanosome brucei, Africa. Trypanosome cruzi, South America. Okay? You can think of, if you go on the cruise, you're going to go to America. Both are transmitted by vectors. African one, brucei, is transmitted by tsetse fly. South American one is transmitted by kissing bug. Both cause systemic disease. Both are mastigophora, protozoa. Both cause systemic diseases with multiple organ involvement. If untreated, both are lethal. There is no vaccine. Both can be treated. Treatment is not sterilizing, meaning that once you tested positive for either of them, you consider to be positive for life. Like you can be fine, you can live a long life, but you cannot donate blood, for example. This, oh yeah, and kind of general symptoms: African, Trypanosome brucei, sleeping sickness, lassitude, lethargy, and if untreated, eventually death. And Trypanosome brucei is probably the most fascinating. It infects smooth and cardiac muscles. If it infects cardiac muscle, it can damage the conducting system of the body, leading to fibrillations. And if it damages smooth muscles of esophagus and colon, it causes, respectively, megaesophagus and megacolon. In both cases, esophagus and colon will lose ability for peristalsis. And if it's an esophagus, people get swollen. If it's a colon, people can constipate. That makes sense? Any questions? Okay. Let's take a break, come back and talk about the fast pace. It's a review. And we're going to start with the innate immune cells. So cells of innate immunity can be divided broadly into two categories: granulocytes and agranulocytes. So first, granulocytes. There are three types of granulocytes: neutrophils, eosinophils, and basophils. Neutrophils' main role is to fight off bacterial infections. For eosinophils, main role is to fight off infections caused by eukaryotic pathogens. And basophils are chief mediators of inflammation. When basophils end up, these cells are found in the circulation. When basophils end up in the tissue, they become mast cells. And I'm going to talk about the role of mast cells a little bit later. Now, another category of cells here are agranulocytes. And among agranulocytes, there are two cell types that mediate the innate immune responses. One cell type is monocytes that are found in the circulation. And monocytes migrate into the tissue towards the inflamed locus. Inflammation forces monocytes to differentiate, to further change into macrophages. So macrophages target virtually anything. As the name suggests, they are big macro eaters, phages. In the second category of agranulocytes that mediates innate responses are NK cells, which are lymphocytes, responsible for early non-specific killing of virus-infected and cancerous cells. Now, what mechanisms those cells can employ to fight off corresponding pathogens? The first one that comes to mind, of course, is phagocytosis. Now, the mechanism that is possible is called respiratory burst. This is when cells produce oxidative chemicals such as hydrogen peroxide or hypochlorite. They also can produce various cytotoxic compounds resulting in cytotoxicity. And neutrophils, specifically, they can stimulate inflammation, just produce inflammatory mediators. Neutrophils can release NETs. NET is an acronym standing for neutrophil extracellular traps. Those are long chains, that's DNA basically of a neutrophil that is released, and it wraps around the pathogens and traps them in a particular place and doesn't let them to go anywhere. Does that make sense? Finally, number 6 is apoptosis. Not their apoptosis, but apoptosis of infected or wrong cells. Okay. So, which one's using more? Neutrophils can use anything from 1 to 5. Eosinophils can do 2, 4. The basophils virtually only 1, 4. Macrophages. Of course, they can phagocytose and they can also cause inflammation. And NK cell, that will be the apoptotic ones. Now, one more thing that I wanted to highlight here, specifically regarding eosinophils, basophils, and mast cells. In cases of abnormal immune response, eosinophils, basophils, and mast cells also take part in allergic responses. All right? So, how does white blood cells fight off infections? So we're going to try and describe the steps of inflammatory response, like overall inflammation. And we're going to start with the injury to the tissue. Okay, so this star represents tissue injury. Now you remember that tissues are permeated by blood vessels. So here are endothelial cells that form your blood vessel wall, and injury to the tissue results in the release of pro-inflammatory cytokines that end up in the circulation. Right? And eventually these cytokines get all the way to the bone marrow. In bone marrow, cytokines stimulate production of white blood cells. And this is called mucocytosis. The white blood cells get into the circulation and eventually end up near the site of injury. How do they recognize that they are supposed to start fighting the infection? Turns out that inflammatory cytokines not only signal bone marrow to increase production of white blood cells, they also lead to vasodilation, bringing more blood to the site of injury. They lead to increased vascular permeability. Do you see those gaps? So white blood cells can easier get into the peripheral tissue. And they stimulate production of adhesion molecules on the surface of endothelial cells. Now, these adhesion molecules, as you can imagine, facilitate adhesion. Initially, let's say it is a monocyte. Attachment, rolling, firm adhesion, and migration of white blood cell into the peripheral tissue. If it's a monocyte, since peripheral tissue is filled with inflammatory cytokines, this monocyte will differentiate into macrophage, and macrophage will follow the trail of breadcrumbs of cytokines to the site of injury. Now, these 5 stages can be named as follows. So number one, it's the attachment. Number 2 is rolling. Oh, now it's better. Rolling. Number 3 is adhesion. 4 is diapedesis or extravasation. That's the fancy term for white blood cell going into the peripheral tissue. And 5, when they follow the trail of cytokines, is positive chemotaxis. Does that make sense? So white blood cells attach, roll, adhere, move into the tissue. And then, if it's monocytes, they become macrophages and destroy infected cells or bacteria, or whatever, dead cells, If it's neutrophils, they will do the same. They will phagocytose, they will release chemicals of respiratory burst nets and stuff. The main difference between neutrophils and macrophages, neutrophils are cellular kamikazes, so they are one and done. They phagocytose and done. When they die, you know what's formed? Pus. Pus are dead neutrophils. Macrophages can phagocytose multiple times. Am I clear? Now, if you would look at what happens here, so first, vasodilation leads to increased blood flow, which leads to redness, right? So we've got redness. Pro-inflammatory chemicals that are released at the site of infection usually also increase local metabolism and local temperature. So we've got fever. Increased vascular permeability leads to plasma seeping into the tissues And when plasma seeps into the tissues, you've got edema. And together, edema and those inflammatory chemicals cause pain. Those are 4 classic signs of inflammation: redness, fever, edema, and pain, or as Romans taught us, rubor, color, tumor, and dole. Okay? That makes sense? Now, plasma washes the crap away into the lymphatics, and coagulation factors that are present in the plasma, they trap microorganisms in place. All right? This is the mechanism of inflammation, which is one of the innate responses. We're good? Next mechanism, I'm not going to put it in there, phagocytosis. It is very well described in the notes. A cell, either neutrophil or macrophage, forms pseudopodia. Pseudopodia engulf bacteria, for instance. Does that make sense? Forming a vesicle called phagosome. Phagosome fuses with a lysosome. Chemicals in the lysosome destroy bacteria. Okay? Good? All right. What I do want to discuss, and to my knowledge, it's rarely discussed in such a short microbiology course. I would like this course to be like 3 semesters. Nobody would want it. No. Is the concept of pattern recognition. In my opinion, this is a really important concept. And this concept answers the fundamental question. Actually, the concept of pattern recognition was awarded Nobel Prize in Physiology and Medicine several years ago. Well, several years, my lifetime, maybe 10, I don't even know. Okay. So here's the story. How does your innate immune system recognize that a particular cell, that something is wrong, that a particular cell doesn't belong? To answer this question, I'm going to use gram-negative microorganism as an example. Okay? Just a garden variety gram-negative microorganism. So here you have gram-negative bacteria. And you remember we just mentioned it like an hour ago, that in the outer membrane of gram-negative bacteria there is endotoxin, lipopolysaccharide. Lipopolysaccharide, bacterial lipopolysaccharide, can be found only in gram-negatives, not in gram-positives, not in fungi, not in human cells. So it's a unique chemical, unique molecular signature of gram-negative toxin. Got it? So, human cells have the receptor that recognizes lipopolysaccharide. Receptor, in this case, called TLR. You don't need to memorize that pair. I'm not going to ask the pair. I'm asking the concept. Okay? So you've got a molecular signature, LPS, that is called, LPS in this case, is called pathogen associated molecular pattern. Not only LPS can be an example of this. For instance, bacterial flagellum is made of a protein called flagellin, which is not present in human cells. It's another example of pathogen-associated molecular pattern, or pattern. DNA in the human cell must be in the nucleus. If it's in the cytoplasm, it's not right. It's not good. Does that make sense? Probably a sign of a virus infection. So, is it pathogen-associated molecular pattern? Oh, absolutely. Human cells do not have cell walls. So peptidoglycan is another example of pathogen-associated molecular pattern. Am I clear? What is TLR4 then? TLR4 has many other receptors that can recognize pathogen-associated molecular patterns. Is pattern recognition receptor. So in essence, pattern recognition receptor, or PRR, on a human cell recognizes pathogen-associated molecular pattern on a cell of a pathogen. Very good. Is that understood? Pay so much attention to this because this concept sometimes causes confusion. Okay. Now, this pathogen-associated molecular pattern and pattern recognition receptor, they will become quite important slightly later in our conversation. So now I'm going to discuss a couple more immune mechanisms that are important for innate immunity. One of them is really interesting and very evolutionary old mechanism called complement. So complement is a system of proteins found in the blood. It's not associated with any particular cell. Okay? Complement can be activated by three major pathways. It can be activated by a so-called classic pathway. It can be activated by a lectin pathway. And it can be activated by an alternative pathway. Now, what activates each pathway? So classic pathway gets activated by antibodies. Activated by antibodies. Bound to the surface of the pathogen. Lectin, look, important, here's kind of a common theme about complement. Something on the surface of a pathogen activates complement cascade. Antibodies on the surface of the pathogen. Mannose, the sugar on the surface of the pathogen that can bind to lectin, the part of the complement, and activate complement by lectin. Right? Or, in case of alternative pathway, surface molecules on the surface of the pathogen. Okay? Does that make sense? Any questions so far? Now, what is happening next? Each activation pathway leads to a cascade, like signaling cascade. But what's interesting about this signaling cascades is that they all converge at the same point. They are different, and I think it's absurd to try and memorize all those steps unless you're working on them. They all converge in a molecule called C3b convertase. So they all come down to the same point. Okay? And what the outcome of this? There are three main outcomes. One outcome is increased inflammation. So this response here becomes amplified. Okay. Another outcome is opsonization and increased phagocytosis. Before we move any further, let's explain what opsonization is. Use the same analogy over and over. Yes. To make food taste better, we add ketchup, we add mayonnaise, we add mustard, right? Those condiments. Complement basically is a condiment for our immune system. There are sometimes macrophages or neutrophils do not efficiently recognize and phagocytose pathogens. Am I clear? If you take a bacterial cell and cover it with a complement proteins, it's called opsonization. Your neutrophils and macrophages will recognize those complement proteins. Am I clear? They'll recognize it. Huh, that looks good. I'm going to eat it. Does that make sense? So opsonization is basically surrounding a bacterial cell and making it more attractive to phagocytes. And finally, the third mechanism here is called membrane attack complex, MAC. Membrane attack complex, especially if you watched a Voltron cartoon, and I gather that based on your age, nobody watched Voltron cartoon. Makes perfect sense. Have you watched? No? Voltron? It's a giant robot that is formed from 5 smaller robots. It's Japanese kind of anime cartoon, which I watched when I was like 9 or 10. It was super fun. So basically, you have several subunits, complement subunits, that arrange like that giant robot and form a pore. Okay? That pore penetrates bacterial membrane, and then water will flow in and out and that stuff and cell dies. Okay, make sense? Awesome. Now, and the last mechanism for innate immunity that I wanted to address here is interference. Now, I deliberately left some space over here because here's what can happen. I'm going to make this cell a little larger. Now, look what we are dealing with here. We have pathogen, gram-negative bacteria, right? That causes some kind of infection. You don't have to make it larger, it's fine. You probably have to make it. Okay? Do you see that? We have infection, and I want to highlight infection. It can be infection in the tissue, or it can be infection in the cell. It can be infected cell, so I can put like a little asterisk here saying that it's infection. Either extracellular or intracellular infection results in the recognition of pathogen-associated molecular pattern by pattern recognition receptor. Does that make sense? That initiates intracellular signaling that goes into the nucleus, and it stimulates interferon genes. Okay? Interferon genes are being expressed, so transcribed and translated. Does that make sense to you? And that causes the release of interferon. Interferon is a protein, water-soluble protein, so it cannot come across the cell membrane. So it has to interact with interferon receptor. That's interferon receptor on another host cell. Does that make sense to you? And what happens as the result of this interaction? The result of this interaction, interferon stimulates intracellular signaling that signals to the nucleus, to the genes, which antiviral or antibacterial genes must be expressed. Does that make sense? So, like, if I walk down the hallway and get bitten by a zombie, so I walk, that's me, I walk down the hallway, I get bitten by a zombie, I pull out my cell phone, and I call, say, Paige, saying, hey, I was bitten by a zombie. So now she can prepare for the zombie attack. Make sense? So interferons are warning signs that cell can produce to trigger some kind of antimicrobial response. Interferons, can this interferon act on the same cell? Sure thing, why not? Can it act on the nearby cell? Absolutely. Can it get into the blood and act on different cell in the body? It can. It's not that often interference of more local responses, but it can do that. Does that make sense? Good? Generally speaking, if you have like an infection in the blood, interferon response will be more robust because just more cells will produce. But it's imperative to have an infection to produce interferon. Now, does every infection result in interferon production? The answer is no. For instance, well, it's kind of complicated, but it looks like coronavirus does not induce particularly strong interferon response in humans. We're clear. Enia, signaling on the same cell you remember from EPT. Autocrine, adjacent cell. Paracrine, distant cell. Good? Questions? Some people try interferon as a treatment for coronavirus, but the problem is, it's like, when zombies attacking you, start calling, it's kind of too late. People try to do it as a preventative, but interferons are really badass. You try to spray, like interferon spray. In the nose, you can do it. But it's really hard to get approval for it because nobody knows how effective. In general, I can tell you interferons, if you manage to give them before you get infected, oh, they wonderful. They really efficient. You never know when you're going to get infected. So, last bit that I wanted to discuss today, to bring all classes at the same point, is to say a few words in the adaptive immunity. Now, I do have some comparison in the lecture notes, and I will do it here very quickly. Some main differences between the innate and adaptive immunity is that it's all in the note. Innate immunity is usually really fast. It's already there. Like, it's there. Like your chemical protections, low pH in vagina and stomach, and dermicidins on the skin, and lysozyme in the mucus. Physical protections like dry surface of the skin or sticky mucus that traps pathogens. Innate immunity is everything of it is there. It can answer like that, but responses are rather non-specific. They usually are local, so their effectiveness is limited. Adaptive responses are slow because you need to train your immune system, but they're systemic, much more targeted, much more efficient. And what's important, they form immunological memory. So your immune system forms what we call immune response. Okay, immunity against a particular pathogen. You cannot have so your inflammation every time is the same, even against the same pathogen. It does not, it doesn't care if you infected with salmonella for 15th time, the immune response to salmonella will be always the same. Innate immune response to salmonella will be the same. Does that make sense? The key concept in adaptive immunity is the concept of antigen. So antigen is a molecule which is large, complex, and foreign. You have to satisfy normally all three parameters to make it an antigen. What do I mean by that? A protein. Large? Yes. Complex? Absolutely. If it's not yours, it's foreign. It's going to be an antigen. Does that make sense? Now, if you take my proteins and give them to someone else in the room, is it going to make it an antigen? We're all humans, so no, same species. If you take a protein from a monkey and give it to a human, that's an antigen, even though we're very close. The more the distance, probably the stronger the response will be. The more alien, the more foreign it is, the stronger the response. Now, what about science? Can a small molecule serve as an antigen? By itself, usually no, but it can be an antigen if you attach it to something big. Like when Spanish conquistadors started to capture America, Indians were absolutely terrified of them. Native Americans were terrified of them because they thought that it's some new species, a person on a horse. Okay, they were like much larger. They didn't realize that those are two different molecules. What about large but simple? Polyethylene. Super large molecules, very simple, cannot be an antigen. Totally foreign, cannot be an antigen. Make sense? Now, antigen, each antigen can be characterized by certain parts of it called antigenic determinants. So this is an antigen. Not like your immune system does not recognize the antigen. It recognizes antigenic determinants. Okay? It can be like that's examples of pretend antigenic determinants of different shapes. Like if you want to describe someone, you can say, oh, this person wears glasses, or this person has a really long nose, or small ears, something like brown eyes. You talk about very specific features. You good? Now, those antigenic determinants, in general, stimulate the generation. They are recognized by immunoglobulins. Often IG, capital I, small g, immunoglobulin. Also often called antibody. So immunoglobulin, typical immunoglobulin molecule, consists of two heavy chains and two light chains. Now, chains are connected to each other by disulfide bonds. I don't really care. It's not super important. Okay. Now, can you see this? So it looks like a fork with two prongs. Does that make sense? And a handle. So prongs are called FAB segment. So FAB segment stands for antigen binding. And quite literally, an antibody here, look, can bind two antigens because it has two antigen binding domains, antigen binding fragments. Those fragments are variable because you need to be able to bind a lot of antigens. That makes sense? This part here called FC fragment, which stands for constant or sometimes cell binding. Why it is cell binding? Imagine that this is not the minute. This is antibody. So this antibody can bind an antigen. Does that make sense? What's the use of it? But look here. What if antibody will label the antigen, like saying, hey, that's bad stuff. And then a molecule of, let's say, I don't know, the macrophage will have a receptor that can pull the antibody inside together with the antigen to destroy the protein. Does that make sense? I came up with a real good story that can serve as an explanation of how this can work. That's a real story. I'm not making shit up. We were working in cultural environment, Russian cultural environment. My kids and I. And long story short, one of our garden tools ended up in the ditch. There's a ditch, some brook that goes in the back of Russian cultural environment. And it's about 4 or 5 feet from the ground to the water surface. There's no way I can get it. So I get the scrawniest kid, the younger one, by his legs. So I'm macrophage, my kid is an antibody. And the garden tool is antigen, so put him down, grabs the tool, pull him up. Okay, does that make sense? So that's basically what antibodies are for, to label and to focus the attention of your immune system on the antigens. Does that make sense? Any questions? So we're going to finish the recording. Next time, we're going to talk about adaptive immunity, the variability of antibodies, and some immune anomalies.